Thank you, Helen. Okay, next we have Dr. Bernard Fox, Chief Laboratory of Molecular and Tumor Immunology at the Earle Chiles Research Institute of the Oregon Health and Science University, and he's going to speak on preclinical models for vaccine therapies.
Thank you. Disclosures. So I was tasked with talking about vaccines and combinations, kind of to provide this high-level overview, but then to look at identifying evidence gaps and barriers to progress, and then some pragmatic opportunities for action to advance the field. I've just got one slide at the end. So when I think about cancer vaccines, I think of the historical perspective, and I think of the ability to prime immunity. And it was really, I think, the work of Brendan Main that I'll talk about, and that concept has stood the test of time, understanding and talking about tumor immunogenicity and then application of adjuvants. So it's been a long time, but in 1975, I just put this up there, that Edward Foley, pictured there, Richmond Prain there, and Ludwig Gross there, received awards for finding evidence that cancer cells have specific antigens that can elicit a cancer-destructive response. And the study that Brendan Main did back in 1957, which is outlined here and I'm not going to go into, but it's a great read to go through those studies. This is why it stood the test of time. They were really carefully done preclinical animal models that identified unique tumor antigens. And this is a summary of his data and lots of others' data that was summarized by Bob Schreiber in Fundamental Immunology a number of years ago. But if you take methylclanthrin, 3-methylclanthrin, which is a carcinogen similar to cigarette smoke, and inject 10 mice with that carcinogen, you develop 10 different sarcomas. If a pathologist looks at it, they all look the same. If you look at gene expression profiling, they have overexpressed common epitopes. However, if you immunize with tumor A, we'll call it tumor A, 100 mice, and challenge 10 of them with tumor A, you protect those mice from a challenge of tumor A. But if you inject the other 90 mice that you vaccinated with tumor A, you don't protect them. The same is true for tumor B. Vaccination with B protects from B, but not from the other sarcomas. So that's a basic concept, that you've got this immunity that you get when you use a whole tumor vaccine, which may be different from other strategies of vaccines. So then immunogenicity as a concept. The way we evaluate immunogenicity classically is to vaccinate animals with an irradiated tumor and then come back typically 2 weeks later, but people do different times, and challenge with a minimal tumor dose. And in this experiment, we did a study where we looked at a dose where you wouldn't get tumor growth in 100% of animals. And then we looked at whether or not it would grow out. And this is using a B16-BL65 tumor that was selected from an original melanoma that was developed a long time ago, but D5 was actually isolated by Suyu Xu at University of Michigan. And when you look at the ability of that tumor to grow in mice that were not immunized or immunized, what you see is the same, essentially. In these open symbols, those are all the naive mice, and these are mice, the naive mice that got 3 times, or 10 to the third D5 tumor cells, 4 of 5 grew out. Here's an immunized mouse. So not only did the same percentage grow out, but they grew out faster, it appeared to grow out faster. So the vaccination did not give you protection. That's the classic hallmark of a poorly immunogenic model. And in our hands, when you use these models, you don't see any of the checkpoint blockade or costimulatory antibodies having effects in our hands. Other tumor models have various degrees of immunogenicity. So if you immunize, and again, this is 304, which is methylclanthrin tumor induced in my lab, if you immunize with that, you protect 92% of the mice. It's a very strong immunogenic model. Other tumors like MCA310, it's less. And in MPR5, again, it's a poorly immunogenic model. You can't get protection from that model. So depending which model you use, you're going to see different results in your combination immunotherapy studies. So Glenn Dranoff and a team of people set out to address this problem. I mean, what can you do to make these poorly immunogenic tumors immunogenic and get an immune response against them? And essentially, this was published in PNAS back in 1993. And what he did is he took this, a variant of B16, BL6 melanoma, and he genetically engineered it with either IL-1 receptor, recombinant IL-1, IL-2, IL-4, IL-5, IL-6, GM-CSF, interferon gamma, TNF, ICAM, or CD2. And then he went back and he vaccinated mice with all those different B16 variants. And what he found was that GM-CSF, shown here, is the one that when he came back and challenged the mice with a non-GM-CSF containing tumor, that essentially 100% of the animals were protected from tumor challenge. So he was able to convert this. You're looking at the days to growth. Here's no vaccine. Here's if you vaccinate with B16. There's really no difference. But you can see the striking difference in animals surviving long-term without tumor. So the concept of what you can do to modify that response. So what it takes to protect or cure mice in the poorly immunogenic models. And what I want to talk about just briefly now is this concept of what is the minimum, from our perspective, what it takes, the role for CD4 and CD8s. But that's a pretty biased approach because I've been a chauvinist for a long time, a T-cell chauvinist, and I haven't appreciated the impact of B cells, much like what Jay Brzozowski presented earlier in his study showing a potent role for antibody. So this idea of potential role for B cells and antibody in preventing metastases and having anti-tumor effects. So there were lots of studies that were done where you would do adoptive immunotherapy with T cells, and you would then deplete the mice with antibodies against CD4. And you would see that you could get perfectly great therapeutic activity in those mice and cure those mice, thinking that you didn't need the CD4s. But in effect, the CD4s were coming back, and they were having an impact on augmenting the survival of either the CD8 T cells or having other direct anti-tumor effects. So this is actually, I think, the very first report to show that if you completely eliminated CD4s in a model system where you didn't get sterilizing immunity immediately, that in this model, if you transfer CD8s that are grown in MHC2 knockouts into MHC2 knockouts, and you look at the lungs at 14 days, they're perfectly clean. There's no mets in them. But even then, all the animals die of tumors that are growing in other parts of the body. So this is the concept that long term, if you're thinking about it from our side, there's got to be some sort of CD4 immunity in place. We don't always look at it. It may not be specific involved in your vaccine or your adoptive immunotherapy. Maybe it's much like some of the things that both Helen and Helen mentioned about survivin and other responses that may develop, that Malcolm actually showed, where you develop sort of this epitope spreading response, and maybe it's that part of a component. So this part I just throw in, this is because I'm not a B-cell person, but essentially this is work from one of my colleagues at Oregon Health Science University, Ann Hill, in her lab, where they actually made a murine CMV viral vector that expressed TRP2. And TRP2, CMV is kind of interesting because it has this inflating immunity. And so the idea is you could get inflated CD8 epitope immunity to CMV, which can continue to grow through your lifetime. But what she found is that if you vaccinated mice with an MCMV TRP2, you could protect 100% of the mice. And if you went back and you eliminated CD4s or CD8s or both, you had no effect on tumor growth. And then she went back to show that she could transfer serum from an immunized mouse and prevent the outgrowth of this poorly immunogenic B16, BL6 tumor. It doesn't work in a therapy setting once tumor is established, but it plays in the role, in this case, a little different from what Dr. Brzozowski was talking about direct anti-tumor effects, but we're trying to evaluate this in terms of what this might have in terms of combinatorial effects against things like metastatic spread. So I think that one of the things that I at least underappreciate, and I think many in the field, is when you think about cancer vaccines, we should be thinking about things that boost not just CD4 and CD8 T-cell immunity, but think about B-cell responses as well. Another area that I wanted to just touch on was this idea of strategies to induce broad sterilizing immunity. And sort of our philosophy, or some of the people in my lab at least, philosophy on why it's important. And so I want to talk about this idea of tumor heterogeneity, the triple E hypothesis, and then I want to talk a little bit about selection of cancer antigens. So I think that cancer heterogeneity mandates that we get broad immunity. And this is a great cartoon that was published by Bert Vogelstein in Science a number of years ago. It's something we all know, but essentially you start with one clone of cancer. Certain cancers are going to be more unstable than others, but as they metastasize, you can have very different genetic makeup of cancers in different metastatic sites. And Charles Wanston, in a beautiful paper in Science that I think accompanied this version, showed that one patient with lung cancer had 15,000 mutations in one lobe, and in another lobe had 5,000 mutations. So how many of those were common, and kind of the potential risks this has for selection bias in terms of figuring out what epitopes you want to make a vaccine against are, I think, going to be profound. The other component here, this classic triple E hypothesis that the immune system eliminates tumors, and if it doesn't eliminate them completely, tumors can escape by either downregulating that gene or eliminating that protein from the immune system to be able to see it. So I think this is another component of why we think we need broad immunity, that if you select a limited number of epitopes to go after in terms of an anti-tumor immune response, you're giving the tumor an option to get away, to escape, and downregulate. And I think while this may be less of a problem in mouse models where the tumor burden is not so great, when you look at larger tumor burdens in patients where it takes longer time to have the anti-tumor effect, it may be even more profound and more of a problem. So as we think about cancer antigens, there will be, I think there is a big debate in the field today about neoepitopes and non-neoepitopes. And there's different classes, and I'll talk about another class in a second. But the NCI invested a lot of time and resources a number of years ago in a project that Mac Cheever led to look at a spectrum of about 75 targets. And they characterized these for things that are important. Therapeutic function. Was there evidence that an immune response against those antigens could mediate therapeutic effects? And they could. Were they immunogenic? Could you make immune responses? Were they specific for cancer? Were they overexpressed for cancer? And they were. So it's actually a great paper if you want to go back and read that. But it talks a lot about some of the epitopes or antigens that, again, both Malcolm Brenner talked about and Helen Heslop talked about briefly in terms of Survivin, SSX, and others. So I put this up there because I think we're kind of, we're limited by what we can look for when we're looking at what immune responses does our body target when we're targeting cancer. And it's sort of this idea of you're looking for your keys under the streetlight. You're using the tools that you have available. When I was in the surgery branch from the 85 to 90, we looked at all the common shared melanoma antigens, kind of to the point that Jim Yollet raised when he asked Steve the question about things like MART and MAGE and GP100. But with the onset of being able to look at mutated epitopes, we're now focusing on that. The problem is we don't have good tools to look at the vast spectrum of targets that are potential antigens in cancer. And so I'll just point out there's this paper that the Society for Immunotherapy of Cancer just put out recently on novel technologies and emerging biomarkers for personalized cancer immunotherapy. And one of the assays that it highlights there is the use of seromics or protein arrays to assess antibody responses. And I'll just point out that there was a paper that came out of Larry Fong's group at UCSF where they had patients that were receiving anti-CTLA-4 and GM-CSF. And they had responder patients on that trial. He used serum from those patients to run protein arrays before and after therapy. And what you do on these is that usually you take serum, you run it against arrays that are spotted with 9,000 proteins, and then you come back with an anti-IgG antibody. And the important thing about it being an IgG antibody is for a B cell to switch from IgM to IgG, it needs to have CD4 help. And if you ask 100 B cell experts, they're going to tell you 99% of them are going to tell you that it's an antigen-specific CD4 cell that provides that help. So the concept is that you can use antibodies, again, as a surrogate for CD4 T cell immunity and possibly CD8 immunity. And you're going to identify, much like Steve had long lists of genes that had mutations in them, you're going to find that there are individual patients that are recognizing a very broad and diverse array of proteins. In this one, Larry Fong found PAC6, which it turned out that he could find about 3% of the cells in the circulation of that patient who responded to have T cell response against PAC6. We found a response against HIBCH, which there were 4 papers in the world literature when we found that antigen. So I think these are kind of questions that are open for our field. Neoantigens, overexpressed antigens, it's going to be a mixture of I think, all these. So I want to take a step back to another class of antigens, and it's work that Jonathan Yedel has done at the NIH looking at things that are called SLPs and DRIPs. So SLPs are short-lived proteins, and DRIPs are defective ribosomal products or misfolded proteins. So SLPs are, because they're short-lived, are degraded rapidly. And they're not available typically for cross-presentation. So they get degraded through the proteasome, they get shunted into the endoplasmic reticulum, where they get picked up by class 1 and are thought to be the dominant epitopes on the surface of all cells. So long-lived proteins, in contrast, are long-lived. They get recycled through the autophagy pathway, where the proteins get degraded and the amino acids get recycled to make new proteins. These are the proteins that when tumor cells die or normal cells die, that are available to be picked up by antigen-presenting cells and to further drive peripheral tolerance. These, we think there's less evidence that they are driving peripheral tolerance. And so while you will have central tolerance against these, the concept, the hypothesis is these are going to be more readily available to be better cancer antigens. So you can collect those, and in fact, this is work that Hongming Hu in our group has done. He blocked the proteasome with bortezomib, shunted these short-lived proteins over to the autophagy pathway, and he uses as a vaccine. And the concept is they get into these microvesicles, which he's characterized to have lectins for things like F-actin and spectrin on the surface that target CLEC9A, which is a ligand on the cross-presenting dendritic cell. And they've also got lots of other things. This is talking about a project we have in the clinic right now, but they've got lots of TLR agonists, they've got 15 DAMPs, they've got lots of antigens. So what he showed is that, and this is an old paper in Cancer Research, but when he compared it to GVAX, which is a GM-CSF secreting vaccine treating animals that have got 3-day tumors, he saw that that was pretty effective. It has a significant effect, but this was significantly more effective. And I'll point out he was using dendritic cells in this study. So now to come back to this figure. So this figure is, I met Prenman as a graduate student, and I think after being in Steve Rosenberg's lab, where we saw the same thing with the TIL studies, that I focused probably more than 20 years doing patient-specific immunotherapies because of this kind of concept, that if you got to make an immune response, it's got to be patient-specific. So I had a PhD student who wasn't smart enough to say no, and he did an experiment. We said, if in fact these short-lived proteins are things that are going to be common, and you could collect them and vaccinate with them, maybe you could shift the immune response and make it more broad. So this is just experiments we reproduced, same as Prenman Main, 304, 309, 310. Cut to the chase. When you use the autophagosomes from these are dark black boxes. This is 95 to 100% protection. The darker shades here, maybe 60 to 80, these are 20 to 40, but all significant protection. So the concept was it worked, but the problem is for the clinical translation, they're all identical mice. So what he went on to do with Sean Jensen as well, it's Chris Twitty and Sean Jensen, he looked in the owl setting. So he set up 3 different mammary cancer models and 3 different strains of mice. And instead of using it as protection, started doing therapy, and he got away from using dendritic cells and just injected the vaccines intranodally in mice. And then because of that last data I showed you, where we saw immunity, but it wasn't good enough, he added OX40 because of work that Andy Weinberg at our institute and Brendan Curdy have been doing developing OX40. We were very interested in adding that. So essentially that's what he did. I'm just going to cut to this. If you use whole tumor vaccines in this model, and this is VALB-C mice, Black 6 mice, FEB mice, dark shade is significant therapeutic effects. You see only one group gives you therapeutic effects, but if you use these autophagosomes, you can see in 8 of the 9 combinations. And so this is now in a phase 2, an NCI-supported phase 2 trial in non-small cell lung cancer and a pilot in prostate. So I want to just go back for some limitations. So gaps and barriers for the mouse. We can't really address a lot of these problems in the mouse model that we can use in humans, where we can use protein arrays, because we don't have these high-density protein arrays in the mice. So now we're stuck doing things like immunoprecipitation and mass spec to try and figure out what the antigens are. Some of the same points that were presented earlier about the mouse in human cancer. Time, tumor growth and age are different, tumor burden, immune selection, microbiome, and different models can give different results. And I'm just going to show you 2 quick slides. So this is work from Amy Moran and Andy Weinberg's lab using combination immunotherapy. There's no vaccine here. It's just, this is OX40 and an anti-PD-L1. And essentially, if you look at the single agents given alone, there's really not much different. This is a very late stage tumor. It's about 10 to 12 days now, tumor growth. So the synergy, the combination of these 2 together is better. This is some work done by Dave Messenheimer, a PhD student in my lab, where he was combining OX40 with anti-PD-1. So I'm just going to take you through this one right here. So this is OX40 by itself. This is the control. This is no treatment. This is anti-PD-1, so no effect on this model. But the combination of OX40 and anti-PD-1 looks like it does worse. And in fact, when you get to the survival data, it is worse. But if you delay the anti-PD-1 and don't do it at the same time, so you can see that you actually get synergy. So this is looking at OX40 and PD-1 where it's given at the same time. This is looking at OX40 and PD-1 where you've delayed the PD-1 a few more days. So should this be driving our clinical trial design, the difference between those two models? And this is a question, I think, for the panel. And we talked about this, Samir and I talked about this in London at the European Medicine Agency's meeting where Mark actually chaired one of the sessions about a month ago. And I think this is important in terms of what we're going to do for clinical trial design, because there are too many things to do. You can't do big studies, but can you do something else? So I'd like to think that we can start thinking about smaller pilot studies of combinations, because how would you develop that OX40 PD-1 combination today based on different animal model data? And so I'm going to stop because the red light's blinking. Thank you.